The bottom-up engineering of artificial cells requires a reconfigurable cytoskeleton that can organize at distinct locations and dynamically modulate its structural and mechanical properties. Here, inspired by the vast array of actin-binding proteins and their ability to reversibly cross-link or bundle filaments, scientists have designed a library of peptide DNA crosslinkers varying in length, valency, and geometry. Engineering synthetic cytoskeletons is essential for the bottom-up construction of artificial cells. Peptide filaments conjoined through DNA hybridization give rise to tactoid-shaped bundles with tunable aspect ratios and mechanics. When confined in cell-sized water in oil droplets, the DNA crosslinker design guides the localization of cytoskeletal structures at the cortex or within the lumen of the synthetic cells. The tunable spatial arrangement regulates the passive diffusion of payloads within the droplets, and complementary DNA handles allow for the reversible recruitment and release of payloads on and off the cytoskeleton. Heat-induced reconfiguration of peptide DNA architectures triggers shape deformations of droplets, regulated by DNA melting temperatures. Altogether, the modular design of peptide DNA architectures is a powerful strategy towards the bottom-up assembly of synthetic cells. The cellular cytoskeleton consists of hierarchical and dynamic polymers that function as scaffolding components of cells and drive vital processes, including cell division, motility, and morphogenesis. Researchers use innovative approaches to build functional cells, bridging the gap between synthetic and living materials. In a new study published in Nature Chemistry, researchers describe the steps they took to manipulate DNA and proteins, essential building blocks of life, to create cells that look and act like cells from the body. This accomplishment, a first in the field, has implications for efforts in regenerative medicine, drug delivery systems, and diagnostic tools. With this discovery, we can think of engineering fabrics or tissues that can be sensitive to changes in their environment and behave in dynamic ways. Cells and tissues are made of proteins that come together to perform tasks and make structures. Proteins are essential for forming the framework of a cell, called the cytoskeleton. Without it, cells wouldn't be able to function. The cytoskeleton allows cells to be flexible, both in shape and in response to their environment. Without using natural proteins, the scientists build cells with functional cytoskeletons that can change shape and react to their surroundings. To do this, they use a new programmable peptide DNA technology that directs peptides, the building blocks of proteins, and repurpose genetic material to work together to form a cytoskeleton. DNA does not normally appear in a cytoskeleton. Researchers reprogram sequences of DNA so that it acts as an architectural material binding the peptides together. Once this programmed material was placed in a droplet of water, the structures took shape. The ability to program DNA in this way means scientists can create cells to serve specific functions and even fine-tune a cell's response to external stresses. While living cells are more complex than the synthetic ones created by the scientists, they are also more unpredictable and more susceptible to hostile environments like severe temperatures. The synthetic cells were stable even at 122 degrees Fahrenheit, opening up the possibility of manufacturing cells with extraordinary capabilities in environments normally unsuitable to human life. Instead of creating materials that are made to last, these materials are made to task perform a specific function and then modify themselves to serve a new function. Their application can be customized by adding different peptide or DNA designs to program cells in materials like fabrics or tissues. These new materials can integrate with other synthetic cell technologies or with potential applications that could revolutionize fields like biotechnology and medicine. This research helps us understand what makes life. This synthetic cell technology will not just enable us to reproduce what nature does, but also make materials that surpass biology. Thank you for watching. If you've enjoyed our videos, subscribe to our channel for there's more to come.